Hello, grade 12 students. Here we go for the last document of chapter 1, document 6, the last process, fertilization. What is fertilization? Fertilization is the union of two haploid gametes, the sperm cell from the male, oocyte 2 from the male, giving rise to the diploid cell, which is the zygote. This process takes place in the upper third part of the oviduct. For successful fertilization to occur, a woman must have abundant thin cervical mucus fluid that will decrease the acidity of the vagina, protecting the sperm cell. Should be in her ovulation day or a day after the ovulation. Should have healthy oviducts and healthy uterus. For successful fertilization, a man must have normal quantity and quality of sperm cell, healthy vas deferens. What are the stages of fertilization? First stage is the sperm cell adherence, the recognition and the attraction process. The motility of the sperm cell is activated whenever they approach toward the oocyte 2. They force their way to penetrate inside the layers of the follicular cells, assisted by enzymes. These enzymes are present in the acrosome, and this is prior to acrosome reaction. Then they are going to adhere to the zona pellucida. Entry of sperm cell, acrosomal reaction, penetration, and absorption. The adherence to the zona pellucida will release these acrosomal enzymes. This process is called, as I said, acrosomal reaction. The action of the enzyme and the propulsion of the flagellum will enable the sperm cell to enter the zona pellucida and move toward something called the periocytal space. It then unites tangentially in a tangent manner to the plasma membrane of the oocyte 2 that will absorb the head. The whole male gamete is going to enter. As soon as this sperm cell will enter inside the cell, the oocyte 2 will be activated to undergo several changes. Cortical reaction, cytoplasmic changes. The oocyte 2 released by exocytosis the cortical granules or vesicles, content which will lead to the formation of fertilization membrane. Pay attention. This fertilization membrane will lead to the modification of the zona pellucida and will become impermeable for other sperm cells. This will prevent what's known by polyspermy, meaning that the entrance of more than one sperm cell. On the other hand, the periocytal space will enlarge in size, yet the oocyte in general will diminish in size. Nuclear changes, resumption of meiosis. The second mitotic division were blocked since the ovulation, resume, and ends after half an hour by the release of the second polar body and the formation of oocyte, which finally will give what's known by the ovum. Formation and diffusion of the pronuclei. The nucleus of the ovum will swell and become the female pronucleus. The sperm will lose its tail and its midpiece, and so it will lose the mitochondria, and the nucleus will swell, forming the male pronucleus. These two pronuclei, the male pronucleus and the female pronucleus, they will fuse in the center of the ovum after duplicating their chromosomal contents. That's why the chromosomes now are made up of two chromatids each. The pronuclei envelope break down and the maternal and paternal chromosome they will mix together. This is what we call by karyogamy. As such, the fertilized egg, which is the zygote, being the first cell of the embryo, is born. You have to know that karyogamy will restore diploidy. It wakes up the female gamete that becomes able to divide.